I want to begin by thanking the organizers for the invitation to speak today. Right off the bat, I want to state that this work was a collaborative effort between the Kumar and Ghosh labs. I also want to acknowledge the people who deserve the kudos for this work, our collaborators, the patients, their families, and funding agencies that supported various aspects of this work. Since the topic of this presentation is prostate cancer, I would like to introduce the prostate to you. The prostate is a walnut-shaped endocrine gland below the urinary bladder surrounding the urethra. It produces seminal fluid, nourishes sperms, and is responsible for urinary and ejaculatory control. Age is the primary risk factor for prostate cancer and is therefore relevant to the theme of this conference. Other risk factors include family history, such as male relative diagnosed with prostate cancer, genetics, such as germline mutations in DNA damage response genes, race, meaning men of African descent, chronic inflammation, and modifiable risk lifestyle factors such as smoking, obesity, diet, etc. The likelihood of developing the disease increases as men grow older. For men under 40, prostate cancer is extremely rare, 0.005% probability. In the 40 to 59 age group, the risk jumps to about 2%, and about 60% of all prostate cancer cases are diagnosed in men age 65 or older, and the average age at diagnosis is 66. By age 80, the chance of developing prostate cancer is nearly 60%. This has implications for treatment tolerability and the appeal for radiosensitizing agents that could reduce required radiation doses. The standard of care for prostate cancer patients with androgen-dependent localized disease is prostatectomy or radiation. Both approaches reduce prostate cancer levels. In many patients, the cancer recurs after a few years. This biochemically recurrent disease is treated with androgen deprivation therapy, also known as castration, since the cancer cells are dependent on androgens for growth. Eventually, the cancer in most patients progress to castration-resistant disease. Plasticity of advanced prostate cancer, castrate-resistant prostate cancer, changes the epithelial cancer into neuroendocrine phenotype, for which currently limited treatment options like chemotherapy are available. These further underscore the need to improve radiosensitization at earlier disease stages. The work that I will be discussing today is rooted in an anecdotal observation that men who took the commercially available Nexrutine pills for pain relief reported a drop in PSA. Next is a bark extract from the cork tree, Philodendron amurans. After initial cell-killing studies in culture, we tested it in a transgenic mouse model of prostate cancer. Dietary intervention with NEX produced a dramatic reduction in tumor volume. Next, we looked at whether NEX could potentiate radiation treatment and found that intervention with NEX prior to RT significantly reduced the number of animals with high-grade tumors. To determine the translational step from bench to bedside, a pilot phase 0, phase 1 window of opportunity clinical trial was designed to evaluate the safety of NEX in prostate cancer patients. Patients with localized cancer received twice daily dose of 500 milligrams of NEX for 2 months prior to scheduled prostatectomy or radiotherapy. Changes in PSA was used to determine evidence for potentiating the effects of NEX in the clinical setting. Biologics were collected at the time of NEX intervention and again prior to surgery or radiation therapy. Although this was primarily a safety study, we were surprised to find that PSA levels dropped significantly in patients that received NEX intervention prior to standard of care treatment. This translational step validated the clinical relevance of the preclinical findings regarding the ability of NEX to potentiate radiation and demonstrated measurable biological effects in patients bridging the gap between preclinical promise and clinical application. Gene expression profiling to compare the combination to single-agent controls led to the identification of several differentially expressed genes. From that set of DEGs, we identified RPS6KB1 as one of the top 8 significantly downregulated gene in the combination treatment. RPS6KB1 encodes for the P70 S6K1 protein, which is a key signaling node. Importantly, it has been shown to be associated with radioresistance. Protein validation using Western blotting confirmed that the combination inhibited P70S6K, its phosphorylated form, and its ability to phosphorylate its downstream target. RPS6KB1 is significantly overexpressed in prostate adenocarcinoma. There was statistically significant increase in RPS6KB1 expression in higher Gleason score cancer relative to Gleason 6, which is low-grade disease. Further, log-rank analysis showed a significant association between high RPS6KB1 expression and poor survival. These clinical data make S6K a biologically relevant therapeutic target for prostate cancer treatment. For more precise pharmacological targeting and dose optimization compared to the crude botanical extract, we conducted activity-driven fractionation of the extract and identified berberin as the most biologically active fraction of NEX. Computational docking showed that berberin fits well into the ATP binding pocket of S6K1 without making any steric or unfavorable clashes with the receptor protein. Further, in a novel mode, phenylalanine 359 from C-class region stabilizes the dimethoxybenzene and benzodioxyl rings of berberin via hydrophobic interactions and hydrogen bonds mediated by the phenylalanine 359 side chain and protein backbone atoms respectively. Western blotting confirmed inhibition of P70S6K and its downstream target RPS6 following treatment with increasing doses of berberin. These results suggest that berberin can potentially serve as a pharmacological inhibitor of S6K1. Prostate cancer cells were treated with escalating doses of berberin and found to inhibit cell survival and their ability to migrate, both of which are hallmarks of cancer cells. Another hallmark of metastatic cells is their high mechanical fitness, such as low stiffness, high adhesion, and deformability, which allows these cells to migrate and metastasize to distant organs. Using atomic force microscopy, we found that berberin treatment decreased the ability to adhere and deform while increasing their stiffness. This means that berberin treatment can decrease the metastatic capability of these cells. Berberin significantly potentiated the cytotoxic effects of radiation in prostate cancer cells. As you can see here, berberin combined with radiation reduces prostate cancer cell growth more than radiation alone. This suggests it may be possible to lower the effective dose of radiation by combining radiation with berberin, which has implications for radiation-induced toxicity. This radiosensitization effect is the core finding supporting the therapeutic strategy of combining berberin with radiotherapy for improved patient outcomes. To determine if these correlative data could be attributed directly to S6K berberin interaction, we knocked out S6K and overexpressed S6K in the knockout cells. While S6K knockout made the cells resistant to berberin treatment, and S6K overexpression restored sensitivity to berberin. The S6K knockout effect on radiation was quite dramatic, as the cells were totally wiped out at a dose of 0.5 gray, unlike the parent or overexpressing cells. These data established a causal link between S6K pathway activity and radiosensitization, confirming S6K as the critical mediator of berberin's radiation-enhancing effects. To validate the in vitro findings, we orthotopically implanted S6K knockout and parental cells in the prostate of mice. After tumors formed, the mice were randomly exposed to radiation or not. Animals were sacrificed at 8 weeks post-radiation. S6K knockout cells formed small tumors in mice compared with parental cells, and they also responded to radiation, forming significantly smaller tumors compared with the radiated parental cell tumors. Circulating tumor cells from the animals bearing parental cell tumors had low stiffness and high addition and deformation, while the S6K knockout cell tumors were stiffer with low adhesion and deformation, suggesting that S6K depletion reduces the metastatic potential even in the animals. These in vivo results demonstrate that S6K depletion potentiates radiation effect in animal models, validating the in vitro findings in a physiologically relevant system. RNA-seq was conducted to decipher the genes that were differentially expressed upon S6K knockout. Heat map of differentially expressed genes shows genes involved in cell cycle, DNA repair, homologous recombination repair, non-homologous end joining repair, NF-kappa B signaling, and autophagy in knockout cells relative to parental cells. From the volcano plot of the top 10 upregulated and downregulated genes in the knockout versus parental LNCap cells, we identified SQ-STM1. SQ-STM1 is involved in triggering degradation of ubiquitinated substrates by autophagy and plays critical roles in various cancer hallmarks, including response to chemo and radiotherapy. After validating SQ-STM1 RNA and protein, we induced autophagy with rapamycin and found that induction of autophagy increased radiosensitivity. Chloroquine, an autophagy inhibitor, inhibited autophagy induction. To determine the ability of berberin treatment to disrupt autophagy, we determined autophagic flux. Immunoblotting showed that in response to radiation, LC32 levels increased with berberin treatment that was further elevated when autophagy was inhibited with chloroquine. Furthermore, under these conditions, clearance of P62 decreased. These data support the conclusion that disruption of protective autophagy may be a key mechanism by which S6K inhibition sensitizes cancer cells to radiation-induced cell death. The decreased SQ-STM1 mRNA suggested that berberin may inhibit transcription of SQ-STM1. Based on putative binding sites for NF-kappa B on the SQ-STM1 promoter and the fact that autophagy and NF-kappa B signaling were among the pathways identified from our transcriptomic data, SQ-STM1 reporter activity was carried out. Berberin treatment reduced P62 reporter activity, and addition of the NF-kappa B inhibitor rescued the inhibition of P62 reporter activity. Knockout cells had lower activated P65 protein and decreased NF-kappa B luciferase activity compared with parental cells. Berberin alone or in combination with radiation reduced protein levels of total and phosphorylated P65, suggesting that NF-kappa B activation may involve S6K kinase activity. These data connect S6K signaling to both inflammatory NF-kappa B and survival autophagic pathways. Integration of RNA-seq data from The S6K knockout cells with parental cells treated with berberin led to the identification of a 39 gene signature of downregulated genes. Low expression of this signature was associated with higher biochemical recurrent disease-free survival. The most significantly downregulated was H2AFX, which encodes for H2AX. H2AX, as most of you know, serves as a beacon for DNA damage and therefore is highly relevant to DNA strand break repair following radiation. H2AFX expression in human prostate tumor is significantly elevated in high Gleason tumors. Log-rank test of disease-free survival of human prostate cancer patients from the TCGA prostate cancer database correlated with low H2AFX expression level. The H2AFX data led us to investigate DNA repair. Using droplet digital PCR, we found that the alternative end-joining repair pathway was significantly reduced in S6K knockout cells compared with the parental cells. Emerging evidence demonstrates that ALT-EJ is an error-prone backup mechanism under conditions of homologous repair deficiency, and Pol-theta, encoded by the POLQ gene, plays a critical role in promoting ALT-EJ-mediated DNA repair in response to radiation-induced double-strand DNA breaks. Polymerase theta has also been shown to be synthetic lethal with DNA repair genes such as BRCA1 and 2 and RAD51, and is seen in advanced prostate cancer. Furthermore, higher polymerase Q expression was associated with decreased survival probability. Gene expression levels of POLQ correlated with H2AFX in prostate cancer patients. We found POLQ mRNA expression decreased with depletion of S6K. S6K overexpression increased POLQ expression. Combination of berberin and radiation significantly decreased Pol-theta protein and POLQ mRNA expression compared with the single agent. Genetic suppression of Pol-theta increased radiation sensitivity, and berberin radiation combination specifically decreased ALT-EJ DNA repair, a mutagenic repair pathway linked to genomic instability. Targeting ALT-EJ through the POLQ axis while simultaneously applying radiation creates synthetic lethality in cancer cells. Our work showed for the first time that elevated expression of a 4-gene signature, namely RPS6KB1, NF-kappa B, SQSTM1, and Pol-theta, is associated with aneuploidy, genomic alterations, and tumor aggressiveness of prostate cancer patients. This study demonstrated the clinical relevance and potential of this signature as a companion diagnostic. This gene panel could help identify patients most likely to benefit from S6K-POLQ targeted radiosensitization therapy. Our working model of radioresistance in prostate cancer shows the involvement of S6K activity-mediated downstream effects that increase efficiency of autophagic flux and double-strand break repair in prostate cancer cells. Therefore, inhibition of S6K activity with a pharmacological agent, such as berberin, makes prostate cancer cells sensitive to radiation through impaired autophagic flux, increased DNA damage, mitotic catastrophe, and cell death. Thank you for your attention.